and I think everybody had time to get a coffee, so I think we can continue with the workshop. So yeah, it's a pleasure to have Dr. André F. Martins here, who is joining us here from Tübingen. He's a local, of course, it's really nice. So he did his PhD at the University of Coimbra, I'm not sure if I pronounced this right, in Portugal, and the University of Orleans in France, where he developed multimodal imaging probes for the diagnostics of Alzheimer's disease, and then moved on to Texas, to the university, and to the Southwestern Medical Center, where he developed innovative MR imaging agents that are responsive to the metabolism. Unfortunately for us, in 2019, he then moved to Tübingen to continue his research at the Werner Siemens Imaging Center at the university to look for biomarkers, to metabolic imaging biomarkers, and among others also for the detection of cancer and the characterization of cancer. And actually this year he received the prestigious Sofia Kovalevskaya Prize for his work. So congrats on that again. And I think we're all excited now for your talk, and stage is yours.
Thank you very much. That was a great introduction. So I feel very humble for all that. So first of all, thank you for organizing this. Can you hear me? Yeah.
We can hear you. It's a bit the audio is a bit not quite perfect, but just continue. I think it's going to be fine.
Okay. So if it's not perfect, you just let me know. Okay.
Yeah.
Now it's good, right?
It's good. Yeah.
So I was saying that, well, very nice introduction. Thank you very much, and for the organization to invite me. This is a great pleasure for me. So hopefully I can show you today some things that are going to be interesting. Well, this morning and even this afternoon, I've seen very great talks about great projects as well. So today I'm going to talk a little bit about imaging tissue physiology, as the title suggests. And so first, I will introduce a little bit. So you have done that already, so thank you very much. So I won't go into details, but basically I will show you a little bit of my pathway. And it's true, while I was in Portugal, in the University of Coimbra, that's where I started all my studies and where I was born. And then I had the opportunity to do and to develop my research studies on the PhD program in France, in the CNRS, that is the Centre National de Recherche Scientifique. And then I had again the opportunity to go to the United States, but not only to the United States, really to the deep America. So Dallas in Texas, in UT Southwestern Medical Center in UT Dallas. And now I had another great opportunity to join you guys in the University Hospital of Tübingen in Germany. And I think, as you can understand, this is like a great pathway. So I still think of myself in Portugal, in the beach, enjoying a little bit of my life. And then from that point on, it was always progressing. And you see, it's life goals. And now I'm in Germany, which is great. And I like it very much. Even though I still sometimes miss a little bit on United States, the Texas barbecue, which is great as well. But well, believe me, this is all made for science. And so I will start actually with this slide, and this is really the beginning of the presentation. And I will also try some things here about questions, so we'll see if that works. So my team actually, when I joined the preclinical department, imaging department in radiopharmacy, also known as Werner Siemens Imaging Center. So I came here actually to lead a team called Hyperpolarized Metabolism and Multimodal Imaging Sciences. And this is just like a big title. I like it, but at the same time, it's too big. And so then we realized that we could actually come with an acronym, and which is now HyperMNMs. And so the HyperMNMs team, what we do is we aim and we are focusing on 4 different domains and another one, but I'm not going to focus here for the sake of the time. So one is imaging the tumor microenvironment. We will see how important that is going to be. Another one is another concept that is the metabolomics, that is studying the metabolism of metals, transition metals, for instance, like zinc, copper, and interesting biogenic metals. Another point interesting for us is metabolic profiling. So it's really using different metabolites to profile, for instance, like different cancer types. And another object of our study and aim as well, a goal, is functional quantitative imaging. And this answers directly a very important question about how to actually do imaging that can avoid false positives and false negatives. And so I start this at this point where imaging is a very broad concept. And basically, when you talk about imaging that is translational or even can be applied into patients, I can name basically 4 that are the most relevant, I think. And so from this imaging, well, for example, magnetic resonance imaging is arguably one of the best imaging modalities for translational studies. Positron emission tomography as well, it's very well known to create great images on metabolism itself. Fluorescence and luminescence is also a great technique that is being applied in surgery or guided surgery for patients. And also, well, the well-known X-ray computed tomography. And so looking at these 4, and there's more, but I focus on these 4, we can see that they are all different, but also they are different in a way that they have different advantages that can be combined together. And for instance, some demonstrate already that they have a very high resolution and temporal resolution, but others have higher sensitivity. But others, for example, optical imaging is very cheap compared to the other, the first 2, and also X-ray computed tomography. But specifically here, so there's 2 techniques that are very, very compatible, and that is magnetic resonance imaging and positron emission tomography, as known as PET, MRI and PET. So PET MRI, let's say. And so I have one question here. So I don't know if this is going to work, but I'm going still to ask, and then I will provide the answer if nobody actually jumps in and answers. So the question is, how can we enhance the tissue and water contrast? And so there's 3 options. The first is we set the magnet on fire, we use a contrast agent, or drink less water. So I'll leave these for a little bit until you think about. You can also give me an answer if you know what it is. I know. And so the answer is use a contrast agent. And so a contrast agent is really used in most of the techniques to enhance the contrast of tissues. And in this case, can also be used for MRI, for example, to enhance the water contrast. I show you here, that's the first imaging that I plan to show you here, because that reflects well the function and how important it is actually to image biogenic metals that I referred before as well. So here in this imaging in the left corner, this is the first imaging that we provided with a sensor that was sensitive to zinc. So we call these the zinc sensors. So this sensor is a contrast agent that basically would enhance the contrast in the presence of zinc. So we applied the first time, so here as you see in this image, in a rat. And so we apply that intravenously. And so what we have seen after injection of the agent plus glucose together, we have seen that something was highlighted. And we didn't know in the beginning what it was, but then we confirmed that was the pancreas. And that's very interesting observation, because it's the first time that we actually see zinc, I mean, the functional imaging of zinc released by the organs itself as a stimulus of glucose. So that reflects as well on the metabolism. And so one of the things that we know now, and I will talk about later as well, is that actually beta cells store insulin inside the cell as a form of granules. And these granules are composed of zinc, a lot of zinc, together with insulin. And this is made actually to protect the insulin to be degraded by other enzymes. And so when we destroyed actually the beta cells in the pancreas with this drug, STZ, we also see that after injection of the sensor, so I'm sorry, this is one in the middle, we see that we don't see any longer the functional effect. So it really reflects that the zinc release was less present in the pancreas, because while the beta cells were destroyed. At the other point, we have a positive control here that you see on the right part of this slide, where we have high-fat diet rats. So they were just fed with a lot of fats for a period of 2 weeks. And then we see that the zinc release now is way enhanced compared to the controls. And so this is a good control that could be used, for example, in diabetes type 2 rats, and also could be translational for patients. So in fact, this sensor now is actually in phase 0 clinical trials. So what happens, as I said, and just to make sure, is that zinc here that is represented in these blue spheres, and insulin that is represented here in this kind of orange sphere, they are inside the beta cells again, as I said. And what happens is that when you have high glucose in the blood, this insulin and the zinc are excreted outside. And what happens is that the zinc normally is not just released outside, it's actually bound to proteins. And one of the most abundant proteins in the blood is HSA, human serum albumin. And that is present in the extracellular space. So what happens is that we have a sensor that goes there and actually binds to the zinc that is bound as well to the HSA. And that reflects on the contrast that is represented with this R1 that is a relaxivity. And that's why we see this enhancement on the images. And this is actually quite functional. So another question, that is another topic that I will present here. And that is, what is the Warbar effect? So if you know, we can answer again. But the question, the answer is cancer cells favor an anaerobic glycolysis. And then actually most tumors that are hyperglycolytic, they overproduce lactate as a product of the metabolism itself. And that happens because basically cells, tumor cells, basically, they are too lazy in a way that they don't have time actually to produce energy into the oxidative phosphorylation or the well-known Krebs cycle. And what they do actually, I'm sorry for this, is that they actually convert glucose to pyruvate and then pyruvate to lactate very extensively. So one of the things that is important is that we know that tumor cells, typically they do that because they just want to create energy in a very fast rate. But one of the things that is not very well known is that actually pyruvate on the conversion goes inside the cell by the transporter MCT1, and glucose as well. So glucose actually can undergo glycolysis and convert it to pyruvate anyway. But the lactate produced that is intercellular actually then has to be secreted outside. And so in our team, we actually think that the extracellular lactate is a better biomarker for cancer detection. And I will try to prove that is true. And so one of the things that we could do here, so there many people actually try to image this lactate, but it's not very easy to do it because as you can imagine, we are talking about something that we have to segregate from the tissues itself. Normally you have intercellular, extracellular, and even inside the membranes, some contributions. And now we are only talking about how to detect extracellular lactate that is outside the cell. There is one technique that is called chemical exchange transfer. And that technique could potentially be used for imaging lactate. The problem here is that you see here, this technique is favored actually for the detection of several metabolites. And one of the applications well known is, and actually is applied in clinics, is glucose test. And this glucose test, because it detects glucose in the tissues by this technique that is chemical exchange transfer. This is a magnetic resonance imaging technique. It's very powerful. And so lactate could be potentially used as well for detection. But as you see here in these red arrows, lactate is very present or is very close to the water peak. So to distinguish in between one and the other is very difficult. But ideally, if you could detect this lactate, you could actually create images that could turn on and turn off in the presence of lactate. And this is actually great. So the question now is, can we actually detect accurately extracellular lactate? So in principle, I would say that, well, only with what we have, it's impossible. And you see here in the right corner, like assessed spectra. This is a typical assessed spectra where we see the magnetization as a function of the chemical shift. And so what you see basically is the water peak is here at 0. And then you basically see our metabolites that will appear differently at different chemical shifts depending on their exchange rates. So lactate alone, we cannot really see it. It's here, but it's really very difficult to distinguish in between, I mean, to separate with the water. But now, when we have lactate in the presence of a shift reagent here, lactate can replace 2 other molecules of this shift reagent or this sensor. And now we can see the europium, I mean, using europium or ytterbium, you can see actually the extracellular lactate present in the solutions. And so to confirm that, so Remy, a student of mine, he just started some experiments where he tried to quantify the extracellular lactate as a measure of CEST, and he has seen that actually this extracellular lactate quantification was quite accurate, as you see here. They're represented here in this red arrow, but then he actually correlated as well with the lactate dehydrogenase assay. And you could see that actually the correlation was almost perfect. So we really could see or detect extracellular lactate by these lactate LDH and also for lactate CEST MRI. So there's an accurate detection of extracellular lactate from cell culture media. He also has done something very interesting, that he chose a phantom where the goal was to create a phantom where we could actually distinguish and profile different cell types and see how they actually could produce extracellular lactate. So we use these cell lines, so PYMT-S2 first generation and the second generation, that is bone tropping. We have here another cell line, MC38, colon carcinoma, and the B16, there is melanoma cancer types. We have in this phantom, so minus is actually reflecting the media with the pyruvate in solution, or an excess of pyruvate, or no, actually no pyruvate, and then the positive, the pyruvate added directly to the solution. So what we have done is that we have also controls here, as you see, PBS, the shift reagent and lactate, and then we have done CEST imaging. And so what we have seen with CEST imaging is that the controls don't show, I mean, with PPF and shift reagent don't show anything, but the control with lactate and shift reagent really showed the presence of lactate. Then we have cells here, the colon carcinomas that showed a lot of extracellular lactate produced by these cancer cells. Then also the PYMT, the breast cancer cells showed lactate produced, that's the second. And then third, the PYMT second generation, there was also different. And second, the melanoma cell lines. There also was it didn't show a lot of extracellular lactate production. And this is very interesting observation because it has been reported that actually melanoma cancer cell lines have very high lactate dehydrogenase B production and expression, which reflects very well what we see as well in the microenvironment. So it really shows that different cell lines can show completely different and distinct extracellular lactate production profiles. And we see here in the graph plot, this is more evident that all the cell lines actually showed completely different extracellular lactate production. But also you see that there's one cell line here that actually showed as well different expression in a way that different production in the presence of not of pyruvate. And that reflects directly to one interesting observation, that is maybe the lactose secretion is rate limited and perhaps is rate limited by different transporters. This is still to be analyzed and confirmed. So we wanted to do that in vivo as well. And so what we have done is that we a mouse model where we implanted some A549. So this is a small cell lung cancers. They are known to be quite aggressive. So we implanted that in a low flank of the mouse. We see here, this is an MRI, a T1 weighted MRI image. And you can appreciate how actually well resolved this imaging can be. We see here the bladder, for example, and you see also the tumor volume depicted here in yellow traces. So what we have done is that we injected our sensor for detecting extracellular lactate. And so what we have seen is this. We have seen that the tumor actually, so the sensor was able to detect extracellular lactate inside the tumor, but it was not the full tumor or the whole tumor itself. It was only a small part of the tumor, and that suggests that the tumor actually is a quite heterogeneous environment, and we are actually seeing now when trying to track into heterogeneity. At the same time, if you do another technique that is another PET technique that is FDG PET, basically uses a tracer that is an analogous of glucose. So what we see is that now the whole tumor is actually highlighted. So it really shows that glucose uptake and the fate of the glucose uptake by the body is completely different. And that suggests as well that determining whether the metabolism or glucose metabolism, what the glucose metabolism does, is really important, determining how heterogeneous is the tumor and what is the fate of aggressiveness of that tumor. And you see here that FDG uptake actually is not as specific as extracellular lactate itself. And so what's the reason for that? So I showed you before that extracellular lactate is important, but we don't see the same for glucose uptake, obviously, because FDG actually, that it should be here in glucose, so we see here, it doesn't go further into glycolysis. So what we see is that FDG goes inside the cell by the GLUT transporter, and then it stays there. And it doesn't do anything else. So we only are seeing the glucose uptake per se. But it's a very complementary technique, as you have seen before, because you really can understand where the tumors are, what they do, how much is the uptake, how glycolytic they are, and then what they do with the glycolysis, the glucose itself. So another technique that we have that is quite important for us is the hyperpolarized imaging. So we have in our center like a spin lab. So what this spin lab does is that it can create metabolic imaging very fast. And this is also a magnetic resonance imaging technique. And what it does is that it can create, actually, you can use metabolites that are labeled with different carbons. So this is a C1 carbon pyruvate. And with that label, we actually can now track metabolism a little bit better in a way that if you have pyruvate, you can look carefully what happens with the lactate version, also alanine, and also bicarbonate. So this is great because now we can really track or try to track like tumor cells and cancers, even in vivo. So the reflection of that is that you got this kind of spectroscopy that is very useful because this is actually 100,000 times enhanced compared to what we would have if no hyperpolarized imaging would be provided. So in a general term, so to get this spectroscopy with non-hyperpolarized imaging, it would take about 92 hours to get this kind of spectra, and this only takes 20 seconds. And you really can see clearly not only the peaks that are represented for lactate and pyruvate, we also can see the dynamics, the kinetics. So how does this reflect? This reflects in this kind of imaging then, because you can convert the spectroscopy into imaging. And you really see that, and this is a collaboration with some people in San Francisco. And we really see, I think this is a good example. So where you see that if you provide hyperpolarized pyruvate into a rat, so you really see that there's clear differences on when, so the tumor normally, this is a prostate cancer tumor model, so a TREMP mouse. And what you see is that when you look at the pyruvate, really it reflects a little bit on the glucose uptake and consumption as well. But really where the tumor is where lactate is produced. So it really reflects how important that can be for the further imaging and stratification of patients. And this is a very translational technique. So the problem of that is that spin lab, as you see, hyperpolarizes, but is dependent on a parameter that is the T1, and which makes it very fast. So the technique is very fast, but also we have to be very fast injecting the tracers because otherwise you will lose a little bit of the signal. And so we actually started not long ago another technique that is deuterium-magnetic imaging, where we, instead of glucose labeled with carbon-13, we now use glucose, deuterated glucose. So we use deuterium. So deuterium is like hydrogen, but it's a little bit more, is heavier. And so we label with deuterium this glucose that is completely fine. So it's like normal glucose and is already applied in patients. And so with that glucose, we now can track a little bit further on what is the glucose or the glycolytic pathway, so glycolysis here, each one of these metabolites. And also we can track what happens in terms of lactate. And we also can track further on the alpha-ketoglutarate and glutamate pathways. This is very important on the to determine whether the amino acid synthesis is a biomarker or not. So what that reflects, this reflects again on spectroscopy, as you see here, so water, glucose, and lactate. And these can be converted into this lyse imaging. So in fact, in the Max Planck Institute with Professor Klaus Scheffler, so we have a project now running So we are able to provide this kind of imaging with patients. So we are starting now the first volunteers with the deuterated glucose, but in principle, so that's what we could provide you. So we see here that there's different kind of imaging in terms of anatomy. But really, when you want to distinguish in between healthy and abnormal tissue, it's when you actually apply the deuterated glucose and you see clearly that the tumor is doing different things with different metabolites. So it's a functional effect. And you really see where the tumor is when you really see where the lactate production was conceived. So I have another question that is, what happens actually when your muscles get sour? And so that's a basic question, but that happens because you are not young anymore. Well, actually, it was faster than me. So it's actually when acid, lactate, and consequent acidification is produced. So most tumors are apoptolytic, as you see. So they produce lactate, and so they acidify the microenvironment. So one of the techniques that we have developed so far is Acidosis. So Acidosis is able to actually track acidity. So what it does is that it uses a compound called iopamidol. And we look here at the kidney. So this is the first standard. This is our gold standard. When we inject this iopamidol in patients, even or in rat models, we see that the kidney can provide two different kind of metabolic shifts. When you convert that, or there's a ratio, that the ratio actually can be converted in a ratiometric image that is actually dependent on the pH. And so since it's dependent on the pH, now we can actually then convert into the pH maps. And this is quite useful. This is the only technique that actually can provide insight on the acidity itself in a very accurate way. So this is extracellular pH measures. And so we have done that. So this technique is developed and implemented in our center. And so what we have done as well, we have some work. So clearly here it shows, you see the kidney, the difference in the gradient from the lumen to the cortex itself. And we have now another examples that we can show. So with this technique, we can now track a little bit more closely to the metabolism. So with Marty Pagel and Sanita Sinare from MD Anderson Cancer Center, so she gladly provided these data for me to present here. We can now see that, for example, when you have tumors that have high expression of carbonic anhydrase and other tumors that have high expression of lactate dehydrogenase, you can clearly see the differences in the pH measures. And not only that, we can also see that now with these Acidosis, we can track very closely, and this is all in vivo data, can track very closely what happens when you have an inhibitor that inhibits the lactate dehydrogenase A. When we apply this inhibitor, now we can see the differences on the pH. Now we have a tumor that is very acidic, which is normal for tumors, normally 6.6. When you inhibit lactate dehydrogenase, then the pH actually is increased, as you see here in the middle at day 6. And then if you stop giving the inhibitor, the pH drops down again to more or less the same level. And this is a great indication that actually Acidosis is able to monitor differences in metabolism. Another concept is a little bit of immunotherapy and acidity. So we actually developed with these two people, and more people actually involved, Manfred and Bernd Pichler. But basically these two, so Philip and Sabrina, actually they developed this a little bit further. And we are now able to see that the pH, or neutralizing the pH, actually favors the way that we actually look now to the immunotherapies itself and the immunoenvironment. And so we actually confirm that this acidity, or this acidity associated with the immunosuppressive environment, is really affecting the way that therapy then can respond or not to different checkpoint inhibitors. And so I think this is the last. I think I still have a little bit of time. And this is the last point where I will talk a little bit about the Sofia Pilovskaya Award and how important actually the QVIC is going to be in this project itself. And so you can think about, this is a very badly representation, but it's the best I could do so far. So this is, I tended to represent like a solid tumor that is well vascularized by blood. And this is, I think, well understood, and arguably I think people can agree with this. And so in this solid cancer, so we normally have several cells, as you see, and vascularization. And you can separate that into actually sample actually the tumor microenvironment. And so what we see here is that when you try to sample with, for example, biopsies, you create like something that you'll see that there's a lot of tumor heterogeneity within these solid tumors. And this is a problem that, I mean, everybody knows, and we are actually getting several approaches actually to determine whether we can image and determine very accurately this tumor heterogeneity. So a good way for me is that I can consider a solid tumor and separate that into actually the cells that compose the tumor itself, and the tumor microenvironment that can involve the fluids that are present in the solid tumor, also the extracellular matrix, immune cells, all that. So it's kind of the soul of the tumor. And so what we intend to do here is that by looking at the heterogeneous tumor, and you know that the tumors are heterogeneous, if you now look at the tumor microenvironment, at the soul of the tumor, maybe you can address a little bit better on how heterogeneous and how specific you can evaluate the tumor itself. And so one of the things we could do is looking at the acidity, as I said before, as you see here. So maybe that can provide good indications and good biomarkers for cancer detection and heterogeneity. The other one could be as well glucose uptake, that is quite complementary. And so we have some evidences as already in the center. And as you see here, we have, this is PET imaging with FDG, the glucose derivative. And you see here that this is a colon carcinoma tumor. And you clearly can see that the tumor uptake already is very heterogeneous. Also, if you use another tracer, for example, FLT, that it's a tracer basically used for detecting tumor proliferation. So if it's proliferating quite fast, so we see that the imaging is a little bit brighter. And so that's what you see here in the same tumor type. So heterogeneity already, you can see that is present here. And we also see that when you take the FDG PET images and we sample different sites on that image, we clearly see that the image intensity is different. But also when you look at the metabolomics, so we take the tissues and then we analyze by metabolomics, it gives completely different profiles. So we really know that the tumor itself that was actually grown within the same cell lines now provides like quite different heterogeneity itself. And this is something that the Cubic actually will help us a lot. And we hope to collaborate even further with them, because now the next step is really to image that and create for each sample, analyze the transcriptomics or the omics itself, and merge together with the metadata analysis from the imaging. So one of the things that we can think about now is that glucose and acidity is not the only thing that we can image. Now we can actually image, for example, lactate, glucose, acidity, and looking at the anatomy and even detect other metabolites. I mean, the imagination is only the limit. So if you have all these imaging together, so now we probably can use machine learning methods to actually cluster the phenotypes on the tumor. And you can think about now, so if you can provide this imaging and understand better how the heterogeneity works, so then we can actually try to modulate the tumor and see whether or not we can now identify the tumor phenotypes, and we can really identify the ones that are more critical to the treatment itself. And so we can approach and you can do what we call precision medicine here, and personalized as well. And so the project itself is really aimed at identify and detect the tumor cancer cells, train and learn, modulate, enhance, create, and because we'll create other therapy models and also sensors, and personalize the therapy itself with the machine learning techniques that we'll develop. So far, I have to thank the team. So this is the team that will provide all these studies and has been creating all this cool science. I'm just a person that is speaking for them, but they are great. And I'm also grateful that the group is quite diverse, as you see here. So thank you very much, this team. Also, thank you very much for the equipment. So this is the most important equipment, and that it's good for you to know as well what we have. So the spin lab is definitely the most important equipment that we have in the lab, as well as several heteronucleic volume coils for metabolic imaging, itself, and functional imaging. We have PET-MR systems, so which is a great environment, and radiochemistry and radiopharmacy associated. So I acknowledge all the people that have been involved in the University of Tübingen so far. So as you see here, as well from the past experience in UT Dallas and UT Southwestern, specifically on the Zinc sensors. Also other collaborators from King's College, Martino Center, and the University of Torino. Stanford University as well, and University of Nottingham, which we start collaborations. And obviously the Cubic that is going to be involved in some of the studies that we have here. And well, I thank as well for Sven for inviting me and all the team that will create great discoveries as well with us. So of course I have to thank to the funding, which is very important, especially to the Humboldt Foundation that really provided and was very generous during these years. And the last but not least, so obviously, so I'm happy to take any questions if you have questions for this presentation, and also later as well if you want. So just please pop me up an email with other questions. And so I think I could convince you here that multimodal in the hybrid imaging is really very powerful and can help understanding metabolism a little bit better. Thank you very much.
Yeah, thanks a lot for this really interesting talk. So it's really fascinating for me. I never knew that you could actually do all these things. So thanks a lot. And yeah, perfectly on time. We have time for a few questions, and there are actually a lot of questions, I think, from, I think Luis had raised his hand first. So Luis, if you want to just pose your question.
Yeah, hello.
Hello, Luis.
Very nice talk. Thank you. I have a small question about, you mentioned the integration of imaging data with omics data. That sounds quite interesting. So this is something, of course, that that it's quite challenging to take samples and really correlate them, especially. Do you guys have any idea what type of structures would you need to integrate this data and be able to analyze it, be able to put it into machine learning methods and so on?
Yeah, that's probably one of the greatest challenges that we will have to face in the next years. But we are confident in a way that we started already 8 years ago. So we have already kind of a setup that makes us like a little bit confident on a way that we will provide that. So we have been publishing as well some data on how to actually analyze first histology. So how we can predict actually how histology can tell us that different clusters can be identified. So I think over there we kind of we progress a little bit. But now the real question is that how we can translate from microscale to macroscale imaging, and that's a bigger challenge. But that's exactly what I intend to frame here. And so what I try to do now is really try to understand histology, because that's the most important data that we have so far. And then try to provide as much imaging together as I can that can be complementary. So if you have that imaging taken care of and the histology together, so then we think that merging the techniques itself is going to be a little bit easier on the way to understand or clustering these phenotypes. And to do that part, actually, you have a machine here that is a milling machine. And the way it works is that, because obviously, as you understand, the histology and the macroscale imaging is completely different. So you really have to take very carefully the samples. And we have that. So we have a machine that clearly identifies what the imaging tells us. And then you can go there specifically and take the sample that we want. And then you can analyze that sample specifically. And that's the way we think we are going to approach and solve the puzzle. Does that make sense?
Yeah. I think I can start to wrap my head around it. Thank you.
Yeah, thank you very much for the question. And well, feel free to contact me again. Because obviously this is something that we want to approach in the future, and I'm happy to new suggestions.
All right, thanks. I think we have time for one more question, and then André will be available after the breakout sessions, right? So
yes, I will. Yes.
I think maybe Sven, I think you had a question.
Then I step back. So I just want to say thank you, André. That's really exciting time, and of course we're very much motivated to bring the omics and the imaging worlds closer together.
Those are magic words. Thank you.
Exactly. Yeah, I have one technical question, but then I ask that later, and I give Philip Riemen Schneider the chance to ask it now.
Thank you.
Okay, great, then I'll just read so Philip had a question, I'll just read it out. So I don't want to sound overcritical, the results are amazing, but I think your imaging greatly depends on the sensitivity, selectivity of your sensors. How can you rule out that this affects the possible treatment of patients?
Yeah, so I think it's worth mentioning that these imaging actually were provided by mice. So the tumors are actually quite small. And so the imaging that I provided here, so we are talking about resolution of about 0.5 micron diameter, which is quite great, to be honest. But indeed, you are right. The resolution and the specificity is also very important. What I can tell you is, for example, the extracellular lactate imaging that we have, that's a quite specific imaging that we can provide. We have tested the sensors with different competitors, for instance, citrate and other things. And for a reason that we still don't understand, these agents are quite sensitive and selective for lactate. And we know that it's extracellular because these sensors don't go inside cells. That's for sure. That's a sure fact. So the challenge here is actually how to differentiate in between, and that's the bigger challenge. Is not really the merging of the techniques. Is really on how we can separate the perfusion from the functional effect that we see. We have some designs in terms of the strategy that we will address, but I think that's a question that it's most of the challenge, because you inject the sensor and then we don't know if the tumor is well perfused or not. And if it's not well perfused, obviously the sensor is not going there, right? And that doesn't happen with hypopolarized imaging, but it happens with the CEST, with the extracellular sensor. Yes.
Perfect. Thanks a lot again. I think we have to move on to our next and final speaker. So thanks again, André. And
thank you very much.